All right, so today we're going to talk about nucleotides and nucleic acids. We have a couple of topics. We're going to focus on the biological function of these molecules, their structure in terms of looking at common nucleotides, and then looking at the structure of double-stranded DNA, as well as ribonucleic acids. And then we'll look a little bit at the denaturation and annealing of DNA, as well as some of the chemistry of nucleic acids. So there are a variety of functions of nucleotides. So energy for metabolism we see in the form of adenosine triphosphate. We see enzyme cofactors like NAD+ or NADH that are electron transport. And then we see signal transduction like CCAMP. And then we also have the storage of genetic information in terms of DNA, the transmission of genetic info like mRNA, as well as ribozymes, and then protein synthesis, the tRNA and rRNA. If we take a look at our nitrogenous base, we also can see that there's a pentose and a phosphate that make up a nucleotide. So there are three components: phosphate, sugar, and the base. And the base is primarily a purine or a pyrimidine. We are familiar already with the functional group, the phosphate, and we just finished studying sugars, so at this point, this pentose should be awfully familiar, especially the fact that the anomeric position is beta with the purine attached. A nucleoside is different than a nucleotide. A nucleoside only has a nitrogenous base and a pentose. The nucleobase is just the nitrogenous base. So either the pyrimidine, which is the single ring shown here, or the purine, which is the two ring. Phosphate group is going to be negatively charged at neutral pH. It's typically attached to the 5' position. So nucleic acids are built using 5' triphosphates. And we also see that nucleic acids contain one phosphate moiety per nucleotide. It can be attached to other positions, but the 5' is the most predominant. If you take a look at some of the other nucleotides, the monophosphate group is actually in different positions. So we've got adenine as our base, and then you can actually see the adenosine 5 monophosphate. You can also see the 2' monophosphate, the 3' monophosphate, and the 2,3 cyclic monophosphate. If you take a look at pentose and nucleotides, the beta D ribofuranose in RNA and the beta 2' deoxy D-ribofuranose in DNA. These are different puckered conformations of the sugared ring. And again, since we know the structure determines function, there's an energy associated with this puckering. If you take a look at aldehyde and the way that it forms beta-furanose, right, this is, again, a reminder, our anomeric position. If we take a look at nucleobases, so these are derivatives of pyrimidine or purine. They're nitrogen-containing heteroaromatic molecules. They are planar or almost planar structures, and they tend to absorb UV light around 250 to 270 nanometers. Cytosine is found in both DNA and RNA. Thymine is found only in DNA, and then uracil is found only in RNA. These are all great H-bond donors and acceptors. Cytosine's pKa at the nitrogen is about 4.5. Thymine's pKa at that nitrogen is about 9.5. These neutral molecules occur at pH 7, so they're physiologically neutral. So adenine and guanine are our purines. Cytosine, thymine, and uracil are the pyrimidines. Adenine and guanine are both found in RNA and DNA. They're really great H-bond donors. The adenine pKa at N1 is 3.8. The guanine pKa at N7 is 2.4. Again, these are neutral molecules at pH 7. That gives you a great place to start when you're thinking about what are the components of nucleotides. Next time we're going to start looking at the structure of DNA.